In this video, we will discuss about UGGT1 and UGGT2, that is UDP galactosyl transferase, and these are important during protein folding in endoplasmic reticulum. As we have discussed this many times about calreticulum cycle in endoplasmic reticulum that takes care of the protein to have to acquire that takes care of the protein to acquire its native form, otherwise it will uh, transfer it to the degradation transfer it for the degradation via ERAD, that is endoplasmic reticulum associated degradation for proteasomal in the cytosol for proteasomal degradation. And now let's come to the topic, that is UGGT1 and UGGT2. And so far we so far people only know that its function is to check whether the protein has acquired its native structure or not, and then if the protein does not acquire its native structure, to reglycosylate it and transfer it again into the calreticulum cycle. And people have identified that UGGT1 works on uh, different kinds of proteins that have versatile lectin chaperones that are works that is it works with different kind of chaperones. uh, However, and before going to another part, and this has been studied using fungal UGGT1 structure, which suggests that the protein sensing domain of UGGT1 has hydrophobic cavity, so it can binds to or it can uh, interact with hydrophobic section of the protein and determine whether it has acquired its native form or not. And it has its UGGT1 has its another paralog, that is UGGT2, and that also has the capacity to reglycosylate. That has been observed by experiment. I'm not going into the experiment about that in this video. However, you can refer the paper that has briefly described about that experiment. So let us understand how the experiment has been conducted to identify the substrates that are that interact with UGGT1 or 2. So in this first panel of the figure, it has shown how the N-glycan, how the initiation of N-glycans occurs. So it happens with the it happens in cytosol in the on the ER membrane with the formation of dolicol single dolicol uh, chain, and then it form into double dolicol and two galactic glycan or sugar carbohydrates added. After that, the 5 mannose has been added, and then it flips inside the ER lumen. There further uh, elongation of this glycan occurs till it acquires uh, triglycosylated 9 mannose and 2 N-acetylglucosamine. So in this enzyme ALGC6 is important for the experiment, as this helps in the addition of first uh, uh, glucose in the glycan in this glycan chain. So if this gene is been knocked out, then this further step would not be able to happen inside the cell. will not be able to happen inside the cell. So they have conducted these studies with ALG6 knockout cell. So how is the experimental design is presented briefly on this second or B panel, and this is the wild type where we can see that the as this 3 glucose mannose 9 and N-acetyl glucosamine 2 structure formed that can bind to uh, signature site of the signature site in the polypeptide chain of N-glycans, that is NXT, asparagine, any amino acid except proline and serine and threonine. However, in ALGC6, what happens? It will not be able to bind. It will not be able to bind to the calnexin calreticulum in the first cycle. However, it will directly goes to the UGGT, which monoglycosylated it. After that, it can bind to the calnexin and calreticulum. And what another thing does in this experiment is that they have added a chemical called DNJ. What it will does is it will inhibit this uh, we can say galactosyl transferase 2 or GST2. That whose function is to trim off monoglycosylated glycan, and when the protein acquires its native structure or release from calnexin and calreticulum, so that UGGT can check whether the protein acquires its native structure or not, and then take further action, such as if it does not acquire its native structure, it will reglycosylate it, or otherwise if it acquires its native structure, it will allow the protein to proceed to the Golgi trafficking. So in this case, in case of this modified this and this GS galactosyl transferase 2 has been inhibited with the help of DNJ protein. Uh, so the monoglycosylated glycan will remain as it is in the modified experiment, and that can be quantified or analyzed in order to understand the insight about UGGT1 and 2 substrate. So what they have done is they have used a recombinant GST-CRT, that is uh, gluthio, uh, glutathione as transferase calreticulum. The calreticulum is been chosen as it can bind to monoglycosylated lectin, and in order to in order to have a uh, control for this GST CRT, they have used GST CRT with a modification, that is amino acid at amino acid at 109 site, that is Y in this case has been replaced with A, and this is considered as the lectin binding site of the uh, GST CRT. So this control will take care of non-specific binding. After that, what they have done is they have trypsin digested it, alkylated, and trypsin digested it, and then do a deglycosylation with the help of PNGSF treatment, and does a mass spec, and then get this figure with the help of some software that has been presented in detail in the paper. But one thing that we need to understand is how they normalize this data or quantify this data is that whatever the TMT levels that they have observed from this GST CRT Y109, and the TMT levels that they have observed from GST CRT, and this one will be divided by this one to obtain a fold increase of the protein in the that has been the interest of this experiment. So what they have identified, they've identified that different kind of proteins that are present in or that has their final destination in different section in the cell are being affected, and is been quantified uh, with this kind of experiment, and this has been presented in the figure C, and more detail are available in the reference paper. And this D figure also shows that the location of the proteins that are affected by ALG6 negative ALG6 knockout cells, and these glycoproteins and this N-glycopropio identify the proteins uh, that are that are affected by GST CRT, or we can say that are affected by, or that has UGGT1 or 2, uh, it's that has UGGT1 or 2 during their protein formation, that needs UGGT1 or 2 during their protein formation. That is a correct word. And they have identified that around 37% are in plasma membrane and 32.1% is not defined, whereas 20% is the major chunk are the secreted protein, and 5% are endoplasmic, are in endoplasmic reticulum, and rest of them can be find out from this figure, and more detail are available in the reference paper. So this is about how they have designed their experiment to find out their ultimate goal, that is the substrates that are substrates that are uh, directly interact with UGGT1 or 2. So that's all for substrates or the protein. You can say that finally it will take the form of protein or glycoproteins. So that's all for this video. Thank you for your time.